All right, so the first batch of research questions that we're going to address. First one, how do we know that DNA is the molecule responsible for heredity? Can cell DNA change within the lifetime of the cell? What holds the two strands of DNA together? And then finally, how does the structure of DNA imply its function? And so again, these are kind of introductory, just what's going on inside DNA that then we can go and build on when we talk about, okay, well, how is it copied and where do we go from here? All right, so how do we know that DNA is the molecule responsible for heredity? All right. First experiment, the Griffith experiment, basically showed that you could, when you mixed dead pathogenic bacteria with a live non-pathogenic bacteria, that they transferred something, that the living non-pathogenic bacteria called the R-type could actually get something from the dead S-type bacteria to turn them into S-type bacteria. So there was something that could actually result in a change in form and a change, what you might call a change in phenotype, based on absorbing something from the environment. And so from there, that's just kind of the foundational experiment. From there, we still don't know what it is that is actually leading to the heredity, the actual transmission of that information. So then you get the Avery experiment. And so what Avery did was built on the Griffith experiment and went in and destroyed all of the protein, all of the RNA, and all of the DNA in various trials to find out what actually is being transferred from dead S-type bacteria to living R-type bacteria to transform them into pathogenic bacteria. And so what Avery found was that it's only when the DNA is left alone that the R-type, the harmless bacteria, can be transformed by dead S-type bacteria. And so Avery was able to provide a evidence in favor of DNA being the molecule responsible from heredity, or for hereditary behaviors, hereditary features. The Hershey-Chase experiment took kind of this to the next level to verify what Avery had found and to see if this is true for all organisms. And so what they did is they took phages, which are viruses that infect bacteria. They fluorescently labeled both the protein coat and the material, the nucleic acid inside of the virus, to see what actually gets injected into the bacteria and transforms the bacteria into virus-making machines. And so Hershey and Chase found that it's the DNA, the nucleic acid within the virus, that actually gets injected and transforms the bacteria into virus-making machines. And so these three experiments illustrated pretty strong evidence in favor that it's DNA that actually determines what the cell makes. That it's DNA that determines even what the cell is, right? Whether it's an S bacteria pathogenic form or a R-type, a harmless form, it's whatever's there in the nucleic acids in the DNA that determines that. And so here are some illustrations of these experiments. Some of them are fun. They're not out of your text. They are out of another text, but you cover these same procedures. And so here is the Griffith experiment. Here's a mouse that was given live pathogenic bacteria, and the mice die. you find live S cells in their blood, determine that S cells are virulent. They are the cells that cause disease. Here are mice given the R-type cells, the harmless cells, and these mice are fine. And you find live R cells early on, and then at the end you find that the immune system of the mice have destroyed all of those harmless bacteria long before the mice actually succumb to any disease. Then you show that mice, when they're given S cells that have been killed, they're given bacteria that are pathogenic, but the bacteria have been killed, that the mice are just fine. And the immune system, again, does a good job of destroying this foreign material. So by the time the end of your time is up, you find that the immune system has destroyed all of those, and determined here that it's only the live S cells that are actually pathogenic. And then so when you give the mice live R cells with heat-killed S cells, that the R cells absorb something from those dead S cells. And what you find out with the Hershey-Chase experiment and the Avery experiment is that what those bacteria are getting from the dead bacteria is what? DNA, right? They're absorbing DNA, fragments of DNA from those dead bacteria. Make sense? Cool. So here's the Hershey-Chase experiment. Again, they fluorescently labeled both the protein coat of the virus and fluorescently labeled the DNA, the nucleic acid, and found that it's only the DNA that actually gets injected into the cell, transforming the bacteria into a virus-making machine. So as long as you have that DNA, that genetic code in there, you can generate all of the machines from that genetic material. All right. So at this point, we know it's DNA. We know DNA exists. We know that it's the molecule responsible for heredity. And then the question is, what are the limits of that, right? What are the limits? Is it only what you would call vertical heredity, where material is passed on to offspring? Or, in another way, can DNA actually change within the lifetime of the cell? Another way of asking this is, are there instances of horizontal gene transfer? And we already know from our Griffith experiment and from the Avery experiment that the answer to that is what? Yes, right? Those R-type, the harmless bacteria, were transformed during their lifetime into pathogenic forms. And so in the Griffith experiment, we see this, that the R-type bacteria, the harmless forms, take on DNA from the virulent forms, from dead S-type bacteria, and actually change within their lifetime and start making whatever it was that the S bacteria were making to make them virulent, capable of transmitting disease. So we call this process transformation, and the bacteria that take on that DNA as being transformed. Right? They were transformed in the Griffith and the Avery experiment from R-type bacteria into S-type bacteria. They were transformed, and again, this process, transformation. How is the Hershey-Chase experiment different? You still had bacteria being changed within their lifetime, right? But how is it different? Were the bacteria taking on DNA from their environment, from dead bacteria? Now, how are they getting that new genetic material? Yeah, from a virus. And so this process is called transduction. Or you can say that the bacteria that changed as a response, as a consequence of that, as being transduced, or this process called transduction. So this is transformation, when bacteria take on DNA from their environment. Transduction, when bacteria are forced to take on DNA from a virus. Okay? These bacteria were transformed. These bacteria were transduced. So what you oftentimes find is that this the viral DNA, and again we call viruses that infect bacteria phages, actually doesn't just end up inside of the cell, but gets incorporated into the DNA of the bacterium, and will remain there as long as that virus allows the bacterium to live. And many viruses do this in human cells as well. In fact, you can look at the human genome and find sections in our genome that look like that they were put there by a virus a long time ago, as a virus got incorporated into our genome. And you know what's kind of cool? Listen to this. This is not made up. This is for real. And I care about it, so I'm going to tell you about it. I don't know if you're going to care about it. The part of our immune system, the gene responsible for making the machinery to fight viruses, looks like it was put in our genome by a virus. Isn't that awesome? Yeah.
So is that like the antibodies that we form?
Well, so antibodies are just kind of a general feature meant to target anything foreign. And so some of those can bind viruses. Many antibodies bind things that are larger, like bacteria. And so you have what's an immune response to the presence of a virus that is separate from antibodies.
Okay.
Yeah.
This could be totally off, but how does so they say some mental health things are genetic. Is that in DNA or things like that pass through?
Some of them, yeah. Yeah, to where a lot of it is a genetic illness that you're just not producing what you need to function correctly. So, certainly so. Yeah. Now, so then what are you asking then? Could we transduce those people by giving them whatever they're missing and helping to correct it? They do that. Not, as far as I know, not with mental health disorders, but they do that with other disorders where they will try to replace whatever you don't have. And usually if they'll do genetic screening, they can find this early on and start gene therapy early. But yeah, you do find gene therapy being used, trying to correct what's wrong. And so, viruses have what are called the lytic phase, when they are forcing the bacterial cell or the human cell, whatever it is, to make viral proteins and make new viruses. And then they usually explode the cell so then they can go and infect new cells. That's called the lytic phase. And then the lysogenic phase is when their DNA is incorporated into the DNA of the host. And it's just kind of hiding out, the period of stasis. But it's good for the virus because those bacteria are going to divide, right? They're going to make new bacterial offspring, and each one of those offspring are going to have the viral DNA in them. And so here's just an illustration of the lysogenic cycle. You can see this viral genome illustrated in blue, been incorporated into the bacterial genome, and every time this bacterium divides, it produces daughter cells that both have the viral genome, and then something can trigger it to move into the lytic phase, where now it pops out of the genome, forces the cell to start making viral proteins and making new viral individuals, at which point it will lyse the cell, blow the cell up, and will go and infect new cells. And so as far as what triggers it to move from one to another varies from virus to virus, and many viruses we don't know what actually triggers them to shift from lysogenic to lytic. Which then makes you wonder if parts of the human genome that look like they were put there via virus, is there some trigger that could then shift them over to being lytic or lytic?
That would be a bummer.
That would be a bummer, right? You're talking about like 5,000 years later, all of a sudden you trigger this virus and you get the Walking Dead.
That's a reassuring thing.
Yeah, you're welcome. I have some other reassuring reassuring thoughts to give you as we talk about this idea of inheritance. All right. So that's, just kind of the background of how we discovered that DNA is the molecule responsible for heredity and how we determined what seemed to be the extent to which DNA can function. Although this is missing from your text, is missing from the text where I pulled a lot of these images and figures is that we're finding more and more evidence of horizontal gene transfer in eukaryotes every day. And so in bacteria, we're like, yeah, I mean, bacteria could take on DNA from their environment. They can have viruses inject that DNA into them fairly simply. But we're starting to find more and more evidence of horizontal gene transfer in eukaryotes as well, including human cells, which is kind of fun. All right. So what holds the two strands together? After we finish this slide, we'll have our lecture break. And again, I still forgot to put the lecture break question in there. I'm sorry. Eventually, I'll remember to start doing this so that when you can look at these slides on Canvas, you'll know when we'll have our lecture break. So what holds the two strands of DNA together? Simplest way to answer that question is just millions of hydrogen bonds. What holds the two strands of DNA together? Surprise, there are two strands of DNA held together. That was just kind of assumed by this slide, didn't actually tell you that. But the two strands are held together by millions of hydrogen bonds. And in many cases, tens of millions or hundreds of millions of hydrogen bonds. Where else do we see hydrogen bonds? Yeah, water, right? Water, hydrogen bonding to other water. We talked about this and what makes water so special, right? It's polar. And so you get hydrogen bonds formed. An individual hydrogen bond is weak, but hundreds of millions of hydrogen bonds are extremely strong. All right? Sound good? Makes sense? Right? One little brother is weak, millions of little brothers not so weak. Wouldn't that be awful to have a million little brothers? Gosh, I had one and it was horrific and remains to be. No, actually we're good friends now, but I mean, it was horrific for him. It was great for me, a great joy. Now, according to what's called Chargaff's rule, there's a limit to how these hydrogen bonds form. Purines have to hydrogen bond with a pyrimidine. And so Chargaff's rule says that the number of purines, which is adenine and guanine, those are purines, equals the number of pyrimidines, thymine and cytosine. Okay? And so those two are equal. However many purines you have the same number of pyrimidines. This isn't saying that there are the same number of A's and G's. It's just this total of A plus G has to equal C plus T. Okay? Does that make sense? Okay. And the reason for that is because, and then taking that a step further, the number of A equals the number of T. This is still part of Chargaff's rule. And the number of G equal the number of C. And the reason for this is a purine can only make hydrogen bonds with a pyrimidine. A purine will not hydrogen bond with another purine. Okay? So A will not hydrogen bond with itself. A will not hydrogen bond with G. A purine must hydrogen bond with a pyrimidine. And a little bit more specifically, since then we have the next specific statement that the number of A equals the number of T, and the number of G, the number of C. What is that an indication? What does A hydrogen bond with? Only T. And what does G hydrogen bond with? Only C. And so A makes hydrogen bonds with T, 2 of them to be specific. G makes hydrogen bonds with C, 3 hydrogen bonds to be specific. Yeah.
So when you first did the experiment, it was like really close, but there's a couple that were off. What caused that again?
Probably that at the very end of the chromosome, you have a single strand. You have DNA that's not perfectly bound with a corresponding strand. And so whereas DNA, looks like this roughly, where you've got 2 strands stuck together and woven around each other, that at the very end you can have things like this, where one of those strands extends out past the length of the other strand.
So it still can only bond.
Yes. Yeah, and so any discrepancy in the number is probably this scenario here, where one of the strands extends beyond the length of the other. All right? Lecture break time. I'm ready. I'm sure you're ready because it's Monday and y'all look tired. And I don't mean like y'all look tired, like, as people are like, you look tired because they don't want to say you don't look good, right? So they say you look tired. You just look sleepy. You look fine. Y'all look great. All right? And so what I'd like for you to do is during this lecture break time, working with the people in your group, I would like for you to design me a DNA molecule that is stronger compared to another molecule you draw that is weaker. Okay, so you're going to draw 2 DNA molecules. So each of those molecules is double-stranded, right? Because the DNA molecule has 2 strands. Okay, so you're going to draw 2 DNA molecules. One that is stronger than the other, and illustrate why it's stronger. Okay?
So like incorrectly draw one and incorrectly draw one?
No, they should both be correct. One's stronger than the other. Okay, so think through that, talk about that with those in your group. Try to figure out from what we've talked about on this very slide, what has the potential to make you a stronger and a weaker DNA molecule. Okay, go ahead and take a couple of minutes starting now.
Alright, girl, you're the new boss.
I feel like every one of our discussions has like someone who's trying to
And it's like, it's coming. One of these days.
One of these days I'm gonna like study up and I'm gonna
Did you read the extra part of my email?
Okay, so.
Which email? On your quiz? Just right now? No. I'll read it. Yeah. I mean, if you want, we could talk about it, but I can also just read your email.
What if?
more Grace Church and teachers assistants.
So I feel like if you had something like running up the middle of it, so like you got this bad boy going on, and it's like connected like this, right? Hey, I guess he wants to see affirmation.
Someone drawing it?
All right, make sure you also take some time in your group to elect somebody that's going to share your idea with the class as a whole.
It's got like good pixels. But what about so instead, you run a piece of rebar up through the center of it. No, rebar. Solid metal. And this has like extras.
All right, do we have a group that wants to share your thoughts with the class as a whole?
Am I just supposed to this girl do this?
Anna, you've got the answer for us?
But I'll share what I think.
Okay.
So would it be thymine and adenine? How would you say that? Those would be the weaker one because
Yes.
there's only 2 bonds?
Yes, they are, right?
The cytosine and the guanine would be 3 bonds.
Yes.
Strong.
Yes. So if you were going to design 2 molecules where one is stronger than the other, the stronger one should have more what in it? You have more hydrogen bonds, and so more of which base pair? G's and C's. And so just for simplicity's sake, I'm going to draw you 2 molecules that are exactly 5 bases long, right? So then the other one is going to have 5. Anyways, so... That was close. It didn't go in.
Oh, it didn't go in?
No, it went
Oh, it
behind
looked
it.
like it went in from my view.
G, 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 C, 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 each of those with 3 hydrogen bonds. And then there's one molecule. The other molecule, A, T, 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 2 hydrogen bonds apiece. So now you have a molecule only 5 bases long. This has a total of how many hydrogen bonds? 10. This has a total of how many hydrogen bonds? 15, right? 5 times 3 is 15. I think you guys can help me out. Yes, thank you. Okay, so only 5 bases long. This would be 50% stronger than this one, right? 15 compared to 10. This is 50% stronger, and it's only 5 bases long. When in reality, your average DNA molecule in a human genome is about 25 million bases long. And so you could generate molecules that are significantly stronger than something else. I guess the most extreme you could get is 50% stronger. But still. All right. So let's think about this. Here's an image of this double helix with your adenine and your thymine, your purine, your pyrimidine, your purine, your pyrimidine, your purine, your pyrimidine. So let's think about this. I don't know, let's imagine you're an organism, right? I know you don't have to imagine too hard to imagine that. But you are an organism that lives in hot pools where the average temperature is 90 degrees Celsius. 90 degrees Celsius, whereas our body temperature is 37 degrees Celsius. So if you live in water that is 90 degrees Celsius, you are going to get into thermal equilibrium with that water, right? Same thing that happens in our bodies. If you get in really cold water, eventually your body's going to lose the ability to keep it warm, and you're going to get in thermal equilibrium with that. It's called hypothermia. Okay, so if you're an organism living in that hot pool, and the fluid in your cells is also at 90 degrees Celsius, what do you imagine your DNA molecules look like? They better be GC rich, because that's going to make them stronger, right? So if you're an organism that lives in a hot pool and your cells are subjected to higher temperatures, your DNA molecules better be GC rich. And this is what you find. When you actually look at the genome of organisms, extremophiles that live in hot pools, their genomes are GC rich, keeping their strands of DNA held together even at high temperatures. Our DNA is not GC rich. It's got a pretty good mix of both. All right. The last of our first set of 4, although we're going to get into our second set of 4 today, so when we finish this, it's not time to go. All right? So how does the structure of DNA imply its function? So something this whole team, Francis Crick, James Watson, Maurice Wilkins, Rosalind Franklin, as soon as they published their paper declaring the structure of DNA as a double helix, they already postulated how it would be replicated. Okay, because as you look at this is basically what they published, that it's this double helix, and that these helices form as these bases hydrogen bond with one another. And then so as they looked at this, they said, well, this could be exactly how it's replicated. Because now if you, say, stripped these 2 strands apart and you wanted to make a new strand, you could only pair a particular base with this C, and that would be G, right? And that's what the strand was beforehand. So looking at the structure gives you an idea of how it's going to be copied. So Maurice Wilkins and Rosalind Franklin, they worked on what's called X-ray crystallography. They basically bombarded structures with X-rays and then looked at how that light diffracted, how the light was spread around, and from that actually determined this double helix structure. So yeah, it is cool, but shining X-rays at something is not super healthy, which is why when you get an X-ray done, the person stands behind a wall and they put that, steel curtain over you. So Rosalind Franklin worked a lot with X-rays and actually died from cancer prior to when they won the Nobel Prize. And at that point, you could not award the Nobel Prize posthumously, and you could not award the same Nobel Prize to any more than 3 people at a time. So it left Rosalind Franklin out. But Francis Crick, James Watson, and Maurice Wilkins won the Nobel Prize for explaining the structure of DNA. So Watson and Crick actually got this whole process started over T one day. I don't remember which one was which. I think James Watson was on the faculty and Francis Crick was a grad student. But one of them was a grad student, one of them was a faculty member. They were just having tea one day and got the idea to start figuring out what the structure of DNA is. And so they were biochemists, and so they kind of got this information, the hydrogen bonds. Maurice Wilkins and Rosalind Franklin got the double helix structure, or rather a helix structure, and then from that they were able to figure out that it was a double helix in which both strands formed lined up bases that could hydrogen bond with those on the other strand. And so some of the things they noticed that the two strands are complementary. That is, if this strand has a T in this position, the other strand is going to have an A. That's the idea of complementary, having what allows the hydrogen bonds to form. But they also noted that it's anti-parallel. And so if you look at an individual nucleotide, you have, I don't want to do that. I'll do this. I'll come back to there. Okay, if you look at an individual nucleotide, we have our deoxyribose, the sugar right here. We have our phosphate group, and we have our nitrogenous base. Okay, if you look at an individual nucleotide, you notice that what's forming this sugar phosphate backbone involves carbon 3 and carbon 5. Okay, you see here's carbon 3. You can tell it's carbon 3 because there's a 3 written next to it. Okay, so here's carbon 3, here's carbon 5. The phosphate attaches the carbon 3 from one sugar to carbon 5 of the next. Okay, and so we would say that this strand goes 5' to 3'. At the very top, you have a lonely carbon 5. You see that? That carbon 5, I mean, it's got its phosphate group, but it doesn't have an entire nucleotide, so it's lonely. Okay, a lonely carbon 5. At this end, you have a lonely carbon 3. Okay, so this goes 5' to 3'. But the other strand, the 5' end is down here, and the 3' end is up there. So not only are they complementary, but they're anti-parallel. Okay, so 5' to 3' goes this direction here, goes 5' to 3' that direction there. So there you would have the lonely carbon 3 at the top, and the lonely carbon 5 at the bottom. Okay, now let's go back to here. Okay, so that's what we mean by the strands are both complementary, facilitating bonds forming between the nitrogenous bases, and they're also anti-parallel. Okay, the free carbon 5 is on the opposite ends for the two structures. And that the structure of DNA itself facilitates how you could actually copy the molecule. Because again, if we strip these two strands apart, we could only get an A to bind with a T. We could only get an A to bind with this T. So if you're putting these new nucleotides in, the only way you could do it is to put 5 A's. And then you've generated a molecule that's an exact copy of what you started with. Make sense? And so they went on to continue to publish about DNA for a number of years. And then Francis Crick actually came up with a cool idea to explain why, even though chemically you should have both right-handed and left-handed sugars and amino acids and all of these things, why you only have one of those. And he said because life didn't originate on this planet. It was actually brought here on a meteorite, and for some reason that meteorite only had the right-handed sugars. Kind of idea. It's an interesting idea. All right, so here's the X-ray diffraction. Again, you shoot X-rays at whatever it is. Doesn't matter what it is, DNA, protein, whatever. You shine an X-ray at it and then study how the light diffracts. And so here you can see gaps where there is no light, and from that you can infer a structure. And in this case, some kind of a helix, some kind of a double helix in which you have two strands round around each other. I could have just waited to actually go forward to here. So here's the anti-parallel. You've got that lonely carbon 5 here, lonely carbon 3 here. On the other strand, your lonely carbon 5 is at the bottom, your lonely carbon 3 is at the top. Why do we care? Why does it matter what the structure of DNA is? Yeah.
Because the structure defines the function.
Absolutely. The structure of everything, doesn't matter what it is, structure always determines function, right? And why do we care about the function of DNA?
Because it's what makes us function.
Right, it's the molecule responsible for heredity, right? So if you want to study what actually enables us to be human, part of that is our genetic component. Or if you want to study likelihood of you passing on some genetic abnormality to your children, right? It's helpful to know exactly how all of this works. Or passing on some genetic, excellence to your children as well, right? Not just genetic abnormalities. Like, you know what? I'm really, really smart. What's the probability that my children are going to be really, really smart?
But that's not necessarily heredity, that's just like learned behavior.
Well, it's both. Yeah, there has to be a genetic component as well. It's not entirely genetic, but there has to be a genetic component as well. All right. So important questions, our next set of 4. How do we know which model of DNA replication is correct? So here we'll talk about some different models that were proposed for not necessarily how DNA is replicated, but under kind of which format DNA is replicated. How does the new strand of DNA get written? Here we'll talk about DNA polymerase and some of the other machinery. What other proteins are involved? Here we'll talk about some additional machinery. And then lastly in this set, why is replication both continuous and discontinuous? All right? We're probably only going to get to the first question in this set. So how do we know which model of DNA replication is correct? So here I'll propose to you 3 models, and we'll mess around a little bit to figure out which one works. So there are 3 models for the replication of DNA. The first is called semi-conservative. So in semi-conservative, each daughter molecule, so you start with our parent molecule here, and then each daughter molecule, which would both look like this, but you'd have 2 copies instead of 1, would have 1 old strand, so what made up part of the parent molecule, and then 1 new strand. This is semi-conservative. Semi-conservative, yeah.
Why'd you call a daughter molecule and a submolecule?
Don't know. I don't know. It's an excellent question. Conservative. So in conservative, your 2 daughter strands, one of them would get both of the old copies, and then one of the daughter strand would have both of the new copies. So each of these would still act as a template for knowing what to write. Bless you. But when they actually came back together, you get your 2 old strands coming back together, right? They're old buddies. They're going to come back together, and your 2 new strands are going to come back together. It's an interesting thought. Although if you do it right, there should be no chemical difference between an old strand and a new strand, right? If everything is done correctly. And then the other one is called dispersive. So that's neither parent strand is conserved. Actually, the entire molecule, let's say this molecule is 25 million bases long. As it's replicated, it's actually cut into pieces and then strung back together at the end. This is dispersive. So neither parent strand is conserved. Each single strand has both new and old pieces. Yeah, again, it's this molecule, if it were 25 million bases long, you cut it into sections, replicate those sections, and then stitch it all back together at the end. And so here's just an image of the one that's actually the correct model, which is semi-conservative. This is the one that was Watson's and Crick's model of DNA replication. And so in here you can see, here's our old strand illustrated in gray. Here's our new strand in red. And as these 2 old strands are split apart, the replication happens to start making the new strands. And so when it gets to the end, your 2 old strands are fully separated apart with their new strands already taking up those hydrogen bonds, okay, or making up those hydrogen bonds. Yeah.
Okay, I didn't quite understand the dispersive.
Yeah, I'll show you right now. Let me show you when you were reading it or just right now?
Both.
Both. Okay. Yeah, I'll show you an image that'll actually show you all three models. And so here's semi-conservative, here's conservative, and here's dispersive. So if it's semi-conservative, when you take one molecule and you make two, each one of these molecules should have one old and one new. And then so when you make a third set, or you replicate it a third time, you have 8 total strands, right? You started with 2. Now you have 8 total strands, only 2 old strands. The rest of them are all new, okay, compared to where you started. In conservative replication, you're going to take this parent molecule, you're going to make 2 daughter molecules. One of them is entirely old, one of them is entirely new, and you're going to keep preserving that entirely old molecule no matter how many replication events you get. You're always going to have this one molecule that's entirely old. In dispersive, the idea is that this is cut into sections. There's some machinery to cut this down so it can handle something that's 25 million bases long in a smaller piece. It's like, how do you eat an elephant? The same way you eat anything else, one bite at a time. It just takes a little bit longer, right? No? You've never heard that? How do you, anyways. And so with dispersive, you cut this into sections, and so what you end up getting is a daughter molecule where here you got both old strands because it was just, again, it's just kind of cut up. Each little fragment acts as a template that then all these fall apart and then get kind of put back together as you do this. And so this dispersive, it just seems like a weird idea of why you would go through the process of cutting this all up. And it's because they didn't understand the machinery involved with the actual replication. They kind of had this idea that there's this machine that just reads a whole section at a time and provides a whole section at a time, rather than a machine that looks and does it one base at a time. And so this kind of comes from this idea of having a machine that reads it several bases at a time. 25 million, that's too big. Can't read that all at once, so we'll cut it into pieces and read small sections at a time. But when you know that the actual machinery actually only reads one base at a time, dispersive doesn't make any sense. Why would you even go through the process of cutting it down into fragments? And so here's the experiment that actually showed that what you get is semi-conservative. And so what they did is they went in and they labeled all of the DNA molecules with the heavy version of nitrogen. Okay? They went in, they labeled all of the molecules with the heavy version of nitrogen. Nitrogen comes in 2 main isotopes, nitrogen 14 and nitrogen 15. And so if you had conservative replication, what you would get is after one round of replication, you would have 2 old strands that have the heavy nitrogen, and you'd have 2 new strands that have the most abundant form of nitrogen, which is the light nitrogen, right? So after one round of replication, you would have 2 bands. You'd have a heavy band, and you'd have a light band. But what they actually found is after one round of replication, you actually get an intermediate weight band, one that has 1 heavy and 1 light. And then after a second round of replication, you get that hybrid band again, 1 heavy, 1 light, and then bands that only have the light. Just like looking here, by the time you get to your third round of replication, you have some molecules that are entirely new, and some molecules that have 1 old and 1 new. All right? Does that make sense? So he showed you don't ever get a time this heavy band just completely disappears and never shows up again. So you don't ever form molecules that have 2 old strands. You get either hybrid strands or you get light strands. And then after multiple rounds of replication, this light strand is going to be really, really small compared to, or this hybrid strand, really small compared to the light strand. All right. We're going to go ahead and end here. We'll pick up here on Wednesday with the second set of questions. If you have any questions, we've got about a minute left of our scheduled class time, and we can talk through those if you like.
What actually makes it like twisted?
What makes it, oh yeah, it's a good question. What makes it twisted like that is are kinks in the shape of the nucleotides. So the nucleotides aren't shaped in such a way where you can actually get them to remain side by side, because the nucleotides are shaped like this.
Okay.
And so when you try to string them together, you can't actually generate a straight line. You could, but it's not very stable. You can form